that you wanted to add by way of introduction before we get started, go ahead, and I will leave it over to you until 6 o'clock.
All right, well, let's get started here. So just to introduce myself briefly, I'm Jonathan McLatchie. I did my PhD in biology at the University of Newcastle in Northeast England. And then I worked for 4 years as a college professor of biology at Sattler College in Boston, which is a Christian school. And then after doing that for 4 years, I moved on to work at the Discovery Institute, which is where I am now. I work as a fellow and resident biologist at the Discovery Institute Center for Science and Culture in Seattle, Washington, where I do work and research, writing podcasts, public speaking engagements like this one, relating to intelligent design, which is a study of patterns in nature that bear the hallmarks of conscious activity, which we'll get into in due course. You can find my personal website at jonathanmclatchie.com. That website houses a lot of my articles, writings, videos, and things of that nature. You can also find the Discovery Institute's Center for Science and Culture website at discovery.org/ID. There's also a blog of the Discovery Institute called evolutionnews.org, where I publish a lot. idthefuture.com is the podcast where I am a frequent guest. You can find all these resources also just at the Discovery Institute website. And you can also contact me at jmclatchie@discovery.org if you have any questions after the presentation that we don't get to address this evening, or you want to talk about anything else. So without any further ado, I also mentioned my organization that I direct, which is talkaboutdoubts.com, where basically this is a service that I launched in 2021, December of 2021, which is basically a service that specializes in mentoring Christians who struggle with doubts in regards to their faith, as well as ex-Christians who want to explore whether there's a rational path back to faith. And so I've been doing this privately on my own personal website since 2016, and I'm very passionate about talking to Christians who struggle with doubts about faith. And one of my primary goals with this ministry is to give people the opportunity to hear and be exposed to the best, most robust arguments and evidences for Christianity before they make their decision to abandon the Christian faith. And so we've assembled a team of around 70 plus scholars in various academic disciplines: New Testament scholarship, philosophy, science, Old Testament scholarship, biblical archaeology, textual criticism, etc. We also have psychologists and pastors, and the aim is to take someone that's struggling with doubts in regards to faith and connect them directly with a leading expert on those topics, who will then connect with them by email to set up a private Zoom call to talk about their doubts with them in confidence. So if anyone would like to talk about doubts or questions they have concerning the veracity of the Christian faith, then please feel free to go over to talkaboutdoubts.com and submit a form, and one of our specialists will be in touch. So without any further ado, let's move forward into this evening's presentation. Let me just lay out some agenda for our time together. And first, I want to lay out what is intelligent design so that we're all on the same page about what we're talking about. Then I'll talk about the genetic code of life and how that points to design. And we'll talk about the irreducible complexity of DNA replication. We'll talk about the cause of circularity problem, and then we'll draw our conclusions, and then we can take some questions. So first of all, then, what is intelligent design? So as I said before, intelligent design is essentially the study of patterns in nature that bear the hallmarks of conscious deliberation, rational, intelligent agency. And so how do we detect design in the natural world, in our everyday experience? How do we go about inferring that something is the product of conscious design? Well, I wonder how many of you would look at this image and infer that this must be the product of design. I mean, how many of you think that someone has come to this cliff face and carefully chiseled this particular formation in the cliff face? Well, I guess probably none of you, because this is very evidently the product of wind and erosion. There's no reason to postulate any sort of conscious activity going into the formation of this cliff face. But nonetheless, it's an incredibly complex and very improbable cliff face. And so clearly improbability and complexity is by itself not sufficient to justify an inference to design. What about this cliff face, though? This is from South Dakota. It's called Mount Rushmore, where we see the four recognizable faces of US presidents that are chiseled onto this cliff face. And we immediately infer that this is not merely the product of wind and erosion over long periods of time, but rather this is the product of conscious deliberation. There's been a rational, intelligent agent that has taken it upon himself to devote a large chunk of his life to chiseling the faces of those four US presidents into the cliff face there on Mount Rushmore. Now, what is it that distinguishes and sets apart this cliff face from the one that we looked at previously? They're both immensely improbable. Nonetheless, we infer that the latter and not the former is a product of design. Well, what's going on here? And at this point, I want to introduce the concept of specified complexity, which is really the underlying basis that of design detection in our everyday experience. So to illustrate, so in order to justify inference to design, two criteria have to be met. So first of all, it has to be incredibly complex, or I'm using complexity in this context as a synonym for improbability. And improbability has to be relative, of course, to the probabilistic resources. I'll discuss what I mean by that in a moment. And secondly, specification. So it has to conform to some independently given pattern, right? Like on the cliff face of Mount Rushmore, we saw immense improbability, and we saw specification, namely that it conforms to independently given pattern, specifically the recognizable faces of four US presidents. Now, let me illustrate this by considering an ATM machine. All right, we've all used ATM machines, and the idea is that you put your bank card into the ATM machine, you dial a 4-digit PIN number, and if the PIN number matches the bank card, you're able to withdraw cash. Now, suppose that you've stolen a credit card from someone and you don't know their PIN number, but you would like to withdraw cash from the ATM machine. So you put the credit card into the ATM machine and you begin to randomly dial 4-digit numbers to see if you could extract some cash from that credit card. Well, usually banks will typically lock you out of the account upon 3 failed attempts at dialing in the PIN number. Now, if you only have 3 trials at guessing randomly the 4-digit PIN number, do you think it's more likely that you will succeed or fail in dialing in the 4-digit PIN number and extracting money? Well, obviously, it's far more, overwhelmingly more likely that you would fail in dialing in a 4-digit PIN number by chance if you're just randomly guessing numbers. On a 4-digit PIN pad, 0 to 9, there's 10 raised to the 4th power of possible ways of dialing in a 4-digit number. So that's 10,000 possibilities. So obviously, if you only have 3 trials, it's overwhelmingly more likely that you're going to fail. And this is what we mean by complexity or improbability. And of course, it's relative to the probabilistic resources. In this case, the probabilistic resources would be the number of trials that the bank gives you before locking you out of your account, perhaps 3 trials. That's the probabilistic resources, the number of opportunities you have to get it right. Of course, if you have 10,000 trials or 1,000,000 trials, is more likely that you're going to succeed in guessing the PIN number by chance. And so obviously complexity or improbability relates to or is relative to the probabilistic resources that one has. And of course, the second criterion is specification, and that is that not any PIN number is going to be able to extract money from this credit card. Although all possible PIN numbers of 4 digits are equally complex or equally improbable, there's only one that is specific to this credit card. And so this is what we mean by specification. It conforms to an independently given pattern. In this case, namely the numbers that are needed to unlock this particular credit card as opposed to any other. Okay. So how then should we think about evidence when we're evaluating whether something is evidence for intelligent design or some other hypothesis or anything else? So think about it this way. Let's imagine a court scene where the detective or the forensic expert comes forward and presents an exhibit, namely the murder weapon that was used to commit a homicide. And he notes that on the handle of the murder weapon was found fingerprints that correspond to your fingerprints, and you're the defendant in this case. So is that evidence that the defendant is guilty? Well, yes, it is evidence that the defendant is guilty. It might not necessarily prove that he's guilty, but it is evidence. Why? Because the presence of the accused's fingerprints on the murder weapon is evidence for the guilt hypothesis, because it is more probable, given guilt than given non-guilt, that his fingerprints would be on the murder weapon, right? And as I said, it might not be conclusive by itself necessarily, but it is evidence, and it tends to very significantly, I think, moves the needle in the direction of the guilt hypothesis. There could, of course, be other evidences that are brought to bear. Perhaps there are tread marks in the mud close to where the homicide took place that match footprints that correspond to the defendant's shoes that he's known to possess. Perhaps there are fabrics that are found close to the scene that match fabrics from his sweatshirt. Perhaps CCTV footage has picked him up within a half-mile radius of where the homicide took place at an appropriate time of the evening, and so forth. Now, you can explain all of these evidences away individually, but you have to scotch tape together a lot of auxiliary hypotheses to make the data fit. Whereas a much simpler explanation in this case would be, well, perhaps the defendant's guilty, and that's why all the evidence seems to point that direction. So this is what I mean by evidence, and what we call a Bayesian as a probabilistic term. And the Bayesian, in Bayesian terms, evidence or piece of data is evidence for a hypothesis if and only if the probability of that data is greater given the hypothesis and given its falsity. And so it's expressed essentially as a likelihood ratio of the probability of the evidence given the hypothesis being true versus the probability of that same evidence given the hypothesis is false. And the extent to which that likelihood ratio is top heavy, that is the extent to which you have evidence for your hypothesis. So going back to our cliff face in South Dakota, this is Mount Rushmore, we see that this is evidence for the design hypothesis, because although it's not necessarily the case that these faces are immensely improbable, immensely likely given the hypothesis of design. I mean, there could have been lots of other potential designs that the agent could have implemented. It doesn't need to be these particular faces. Nonetheless, it's far more probable that you'd observe these faces on Mount Rushmore on the hypothesis of design than it would be on its falsity. And so it overwhelmingly confirms a design hypothesis. So again, we see a beach. This is another illustration. We see a beach with grains of sand, and we don't necessarily infer that this is the product of design. It's a product of deposition from the ocean and so forth. Whereas if we see this, if we see inscribed onto the sand, Mary loves John, then we immediately infer this is a product of design, because this has purpose. We have a purposeful arrangement of shapes, which we know communicates a meaning, namely that Mary loves John. And so we infer design without really thinking twice about it. Likewise, this structure is not the product of wind and erosion, but is rather the product of a mind, because we recognize that this communicates information, is meaningful to us. Okay, now a popular misconception about the nature of evidence is that in order for a piece of evidence to strongly justify a hypothesis, the piece of evidence has to be strongly predicted, given that the hypothesis is true. And this is actually false. This is mistaken. So to illustrate this, consider a cabin. Imagine that you're hiking and you're hiking on a mountain. You're far away from roads and civilizations, and you come across this cabin, and you wonder whether the cabin's inhabited. And so you decide to inspect. And so as you prise open the door, you find, lo and behold, this cup of English breakfast tea that is sitting on the counter, still steeping. It's not at room temperature. Now, if the cabin is inhabited, would you predict strongly that you'd find a cup of specifically Earl Grey or English breakfast tea sitting on this countertop? Well, no, not necessarily. And there could be coffee, for example. It could be another type of tea. It could be Earl Grey or chai tea, or there's lots of possibilities. Nonetheless, it's not highly probable, supposing the cabin is inhabited, that you'd make this observation. Nonetheless, it's still far more probable that you'd make this observation on the hypothesis the cabin's inhabited than it would otherwise be. And so it strongly confirms the inhabitation hypothesis. So hopefully that makes sense. You can find if you want to do a deeper dive onto the basis of design detection, I'd recommend these books. This one that just came out recently, the second edition of The Design Inference by William Dembski and Winston Ewart. Another book is No Free Lunch, which is a sequel to the first edition of The Design Inference by Bill Dembski. And then thirdly, I'd recommend Signature in the Cell, DNA and the Evidence for Intelligent Design by Dr. Stephen C. Meyer. So let's move on then to look at biology specifically and try to elucidate why biology points so forcefully towards the conclusion of design. So let's begin by looking at the genetic code of life. So I don't know how many of you have a biology background, but let me just give you a little primer on some basics here. So this is the structure of the DNA double helix, which is hopefully recognizable as a double helical structure. DNA, of course, undergoes replication to prepare for cell division. But in order for DNA to be expressed as a protein, because the sequential arrangement of, so basically DNA is essentially comprised of a sequence of chemical subunits that we represent with the letters A, C, T, and G, which will determine in turn, the sequential arrangement of the subunits of proteins that we call amino acids, right? So basically, you have a language convention of DNA that will specify how the sequential arrangement of the amino acids that form a protein, which will in turn determine how the protein will fold into its three-dimensional structure. And so in order for DNA to be expressed, it is first transcribed. There's an enzyme called RNA polymerase that transcribes DNA into messenger RNA. And then the messenger RNA exits the nucleus and goes to a two-part chemical factory called a ribosome, where it is translated into protein. And the proteins perform various jobs in the cell. So for example, molecular machines in the cell are comprised of different proteins that all have to be very specifically crafted to fit together and achieve some higher level purpose or objective. There are biochemical pathways that are accomplished by a series of enzymatic steps enzymes are essentially chemical catalysts that speed up a chemical reaction, and they're able to convert substrates into products. And so biochemical pathways depend upon proteins that have to be very specifically crafted to fit into their respective substrates and perform catalysis. Okay, so proteins are very important for the cell, and they're encoded by the DNA. And this is what we call the central dogma of molecular biology. Now, I'm going to show you a short animation that reveals how this intricate process works, how you go from the information in DNA to a protein that can perform a job in the cell. So I'll play this animation. We just check my sound is shared. No.
In 1957, Francis Crick first proposed that chemicals called bases along the spine of the DNA molecule function as alphabetic characters in a written language or digital characters in a machine code. This animation shows how this digital information directs protein synthesis. First, a large protein complex separates the tightly wound strands of the DNA to prepare it to be copied. During this process of transcription, a protein complex called a polymerase produces a single-stranded copy of the original instructions. Here we see this copy, a messenger RNA molecule, being constructed inside the polymerase as individual bases are positioned and added to the growing strand. Now we see the polymerase in action from the outside as it spits out the messenger RNA transcript. Next, this RNA transcript approaches and passes through a molecular machine called the nuclear pore complex, an information recognition device that controls the flow of information in and out of the cell's nucleus. Now we see the genetic assembly instructions on the messenger RNA approaching and arriving at a two-part chemical factory called a ribosome, the site of protein synthesis. As the messenger RNA transcript passes through the ribosome, the process of translation begins. During translation, a mechanical assembly line builds a specifically sequenced chain of amino acids in accord with the instructions on the transcript. These amino acids are transported from other parts of the cell by molecules called transfer RNAs, which link specific sequences of bases to corresponding amino acids. The sequential arrangement of the amino acids determines the type of protein constructed. When the construction of the chain is complete, it is transported to a barrel-shaped machine that helps fold it into the precise shape required to perform its function. After the chain is folded into a protein, it is released into the outer cytoplasm to do its job in the cell.
So as you can see from the animation, there is an incredible information processing, storage, and retrieval apparatus in the cell. DNA codes for proteins. DNA is transcribed into messenger RNA and then translated into proteins that will perform various jobs in the cell. And in order for the DNA to specify amino acids, which form the subunits of proteins, there has to be a language convention. And this is the language convention that we see on the screen here. So there are 64 different words, if you will. These are known as codons. And each word is 3 letters long. And each of these codons will specify an amino acid. There's 20 different amino acids. And so there are amino acids that are coded for by multiple codons. There is one start codon. It's AUG, which codes for methionine, which is almost always the first amino acid that is found in any protein. And there are 3 stop codons, UAA, UAG, and UGA. And these basically signal the ribosome to terminate translation. And so we see that there is an incredible language convention that requires explaining. We need to give an explanation for how this language convention came about. And in every realm of experience, we habitually associate language conventions and codes and specified complex information of this sort and information processing, storage, retrieval systems. We habitually associate that with design. And so when we find information content in the cell, language conventions, and information storage, processing, and retrieval apparatus, the best explanation I would suggest is that it too arose by virtue of an intelligent cause, particularly in virtue of the fact that these sorts of phenomena are not particularly surprising, supposing that a mind is involved in their origins, because we associate these sorts of phenomena with conscious activity all the time. But on the other hand, they become wildly surprising on the falsity of the design hypothesis. And so you have this overwhelmingly top-heavy likelihood ratio that in fact supports that these are in fact the product of design. Now, we can go further than that, though, because not only do we have this incredible language convention, but it appears to be very delicately, finely tuned and optimized so as to minimize errors. So the genetic code found in nature is exquisitely tuned to protect the cell from the detrimental effects of substitution mutations. The system is in fact so brilliantly set up that codons that differ by only a single base or a single letter either specify the same amino acid or an amino acid that is a member of a related chemical group. So the assignments of codons to amino acids is non-random. So in other words, the structure of the genetic code is set up to mitigate the effects of errors that might be incorporated during translation. Because if you have two different codons that differ by only one base, the odds are that you're going to get the same amino acid or a very similar amino acid. There are also what I call encrypted stop signs. So another astonishing feature of the genetic code is that the sequences of stop codons overlap with those of the codons specifying the most abundant amino acids. So abundant amino acids like glutamine or glycine have codons that differ by only one base with stop codons. And that is striking because it means that the genetic code is set up in such a way to dampen the impact of what we call frame shift mutations. So what's a frame shift? So a frame shift is where you have an insertion or deletion into the genetic code that is non-divisible by 3. And so that causes an entire reading frame to be shifted. And so the idea here is that by having the most highly used amino acids overlap with the stop codons, is that when you have these frame shift mutations, the odds are that you're going to get stop codons arising and therefore stop translation before it does any damage. So that's another incredible design feature of the genetic code. So there's this paper by, I can't pronounce the name, but it's Kovitz and Allen, which is titled "The Genetic Code is Nearly Optimal for Allowing Additional Sequences Within Protein Encoding Sequences." And they conclude that in the conventional genetic code, translation of a frameshift error is halted faster on average than in 99.3% of alternative codes. And related to this is the ability, also reported by the same paper, of the universal genetic code to allow arbitrary sequences of nucleotides within coding sequences much better than the vast majority of other possible genetic codes. Here's another paper on this subject by Wickman and Ardern. Optimality of the standard genetic code is robust with respect to comparison code sets. There is a whole host of papers in the mainstream academic literature on the incredible optimization and fine-tuning of the genetic code. Here's another paper titled "Did Evolution Select a Non-Random Alphabet of Amino Acids?" And they note that, and I quote, "The standard alphabet exhibits better coverage, i.e. greater breadth, greater evenness than any random set for each of size, charge, and hydrophobicity, and for all combinations thereof." In other words, within the boundaries of our assumptions, a full set of 20 genetically encoded amino acids matches our hypothesized adaptive criterion relative to anything that chance could have assembled from what was available prebiotically. Here's another paper that came to a similar conclusion. They said, "We defined an adaptive set of amino acids as one whose members thoroughly cover relevant physical chemical properties or chemistry space. Using this metric, we compared the encoded amino acid alphabet to random sets of amino acids. These random sets are drawn from a computationally generated compound library containing 1,913 alternative amino acids that lie within the molecular weight range of the encoded amino acids. Sets that cover chemistry space better than the genetically encoded alphabet are extremely rare and energetically costly. Further analysis of more adaptive sets reveals common features and anomalies, and we explore their implications for synthetic biology." So again, incredible evidence of fine-tuning of the genetic code, that language convention system whereby codons translate into amino acids, as well as the actual set of amino acids that's used in biology seems to be highly optimal and not really plausibly the product of chance. Now, could you evolve over time a genetic code and basically arrive at this by chance and trial and error type of process? Well, no, the answer is probably not. So changes in codon assignments would be catastrophic to the cell because such a mutation would ultimately lead to changes to the amino acid sequence in every protein produced by the cell, right? So you can't really evolve over time a genetic code. So, there's this is a significant problem that confronts naturalistic explanations of the origins of this incredible fine-tuning of the standard genetic code. Again, this is not very surprising if design is involved, but is wildly surprising otherwise. Let's then look at the irreducible complexity of DNA replication. So this is the mechanism by which DNA is replicated in preparation for cell division. So I don't know if you've heard of this term irreducible complexity before, but this basically goes back to Charles Darwin in The Origin of Species, which was published, at least his first edition was published in 1859. And he says, if it could be demonstrated that any complex organ existed which could not possibly have been formed by numerous successive slight modifications, my theory would absolutely break down. And in 1996, a biochemist from Lehigh University, Michael Behe, published a book called Darwin's Black Box, in which he sought to take up Darwin's invitation and challenge. He said, and he coined this concept of irreducible complexity, and he defines it as follows. He says, by irreducible complex, I mean a single system composed of several well-matched interacting parts that contribute to the basic function, wherein the removal of any one of the parts causes the system to effectively cease functioning. And so to help us understand the concept of irreducible complexity, he offered as an illustration the mousetrap, which we've all seen. And a mousetrap is comprised of a number of different parts. You have a platform, you have a spring, you have a hammer, you have a hold down bar, you have a catch, you have staples that hold the trap together. And if any of these parts is missing, you don't have a trap that catches half as many mice as it used to, or a quarter as many mice as it used to, but it's broken. And so, and if indeed the trap might still not work if the holding bar is too short or the spring too lightweight. So the parts have to be very carefully matched one to the other and fitted together in order to perform this higher level purpose, in this case of catching mice. And so by product of chance and physical necessity, where you have this step by Darwinian step, where natural selection preserves those combinations that help the organism survive, that confer a benefit to the organism. How would you select for a system like this? By numerous successive slight modifications, retaining utility, an advantage, a benefit at every step along the way. It seems highly unlikely. And indeed, there are many such cases in the biological realm, and we're going to talk about one of those just now. So why is irreducible complexity evidence of design as opposed to chance and physical necessity? Well, explaining the origins of irreducible complex features requires a cause that possesses foresight. And only intelligence is known to be able to visualize a complex higher level objective and bring together everything needed to actualize that end goal. Unguided mindless processes can't do that. And so anything that is going to require foresight to bring about is going to require intelligence, which is the only known cause in the universe that has that ability to plan for the future. So William Paley, who is an intellectual hero of mine, particularly for his biblical scholarship, though he also published a more famous book called Natural Theology, and he puts forward this famous illustration. He says, in crossing a heath, suppose I pitched my foot against a stone and were asked how the stone came to be there. I might possibly answer that for anything I knew to the contrary, it had lain there forever. Nor would it perhaps be very easy to show the absurdity of this answer. But suppose I had found a watch upon the ground, and it should be inquired how the watch happened to be in that place. I should hardly think of the answer I had before given, that for anything I knew, the watch might have always been there. There must have existed at some time and at some place or other an artificer or artificers who formed the watch for the purpose which we find it actually to answer, who comprehended its construction and designed its use. And how vindicated Paley would feel if he were living today in light of our modern advances in the life sciences. I'm going to give you an example which I think is not at all dissimilar from the example that Paley offers us of a watch upon a heath. So when we look at the machinery of DNA replication, we see incredible intricacy and very well-matched parts, an assembly of well-matched parts that are contributing to a higher level purpose of replicating DNA. We see error correction, proofreading mechanisms, and incredibly choreographed system. Here's a short animation, it's only a minute long, that reveals this intricate process.
DNA replication is carried out by a molecular machine called the replisome that pulls apart the double helix and makes an exact copy of each strand. In this visualization, we're going to examine the mechanism for copying DNA. The enzyme DNA polymerase synthesizes DNA molecules, using the original molecule as a template for the production of a complementary strand. The genetic code in the template strand is paired to free-floating bases, matching cytosine to guanine and adenine to thymine. DNA polymerase creates an exact copy of your DNA code, making less than one mistake in a billion bases.
So it's pretty incredible. As you saw in the animation, DNA replication occurs in what we call a semi-conservative manner, which means that each of the strands of the DNA is used as a template for the synthesis of a new complementary strand. And so you start off with one DNA molecule and you end up with two. DNA synthesis is carried out by a group of proteins that act together as a replication machine. And if any of these parts is missing, you don't have DNA replication that works half as well as it used to, or a quarter as well as it used to, but it's broken. And each part has to be very specifically crafted in order to perform its specific job. And so, for example, suppose that you didn't have the topoisomerase enzyme. Now, what does topoisomerase do? So topoisomerase is an enzyme that is responsible for alleviating supercoiling that might be incurred as a result of the torsional stress during copying. And think about, an old telephone cord that ends up becoming supercoiled. And that can happen with DNA as a result of the torsional stress. And this, if left unchecked, would cause the DNA to break during copying, and we don't want that. And so topoisomerase is absolutely crucial for successful replication. In fact, here's a short animation of the topoisomerase enzyme at work.
Wouldn't it be amazing if there were a device that untangled knots automatically? Inside your body, there is. It's called a topoisomerase. It untangles knots in our DNA in an incredible process that keeps us all alive. DNA carries assembly instructions required for your body to generate new cells. But for DNA to do its work, it must be copied in a process known as replication. During replication, the DNA strands are separated, and protein machines use each strand to make a copy of the opposite strand. The end result is 2 copies of the original DNA. However, DNA replication faces a life or death challenge before it can complete its mission. Separating the DNA strands causes twisting in the portion of DNA that has not yet been separated. Eventually, the tension from the twisting increases so much that the uncopied segment of DNA wraps around itself in what are referred to as supercoils. If nothing intervenes, this buildup of supercoils will completely shut down the DNA replication process, since the 2 strands can no longer be separated, and your cells will die. Enter special proteins called topoisomerases to the rescue. Topoisomerases are amazing molecular machines that untangle knots. They come in 2 main types. Type 2 typically has 3 main sections: an upper gate, a middle gate, and a lower gate. Each gate can open or close during the protein's operation. A type 2 topoisomerase untangles DNA in 4 main steps. First, after 2 DNA segments enter through the top gate, it uses the middle gate to break one segment of DNA apart. Next, it passes the other DNA segment through this break. Then occurs the crucial step. The topoisomerase recombines the first DNA segment. The result is the elimination of 2 supercoils. Finally, now that the 2 DNA segments have been untangled, they are released by the topoisomerase. The second strand is released out the bottom and the first strand is released out the top. Of course, this is just a simple summary. The actual process is a lot more complicated. Get ready for a more detailed version of how this process may take place based upon the latest research. When we zoom in on the upper part of the topoisomerase, one of the 2 overlapping DNA segments enters the upper gate. It then binds to the middle gate. Now the second DNA segment enters the upper gate. The DNA in the middle gate is cut in 2 by the topoisomerase. 2 ATP molecules attach to the upper gate and the gate closes. One of the ATP molecules is broken apart into ADP and phosphate, releasing energy that may help keep the upper gate together during the next steps. The middle gate now opens and pulls the 2 halves of the broken DNA apart, creating a gap. Since the middle gate is connected to the broken ends, the DNA remains attached to it. The second DNA segment now moves through this gap. The middle gate closes and the attached DNA ends are reconnected. The upper gate rotates, which may prevent the second segment from moving back through the break. The remaining ATP molecule breaks apart, and the lower gate opens, allowing the second DNA segment to leave. Then the lower gate closes, the upper gate opens, and the first segment of DNA is finally released. The topoisomerase is now reset, ready to perform the same process all over again. The topoisomerase molecular machine is a wonder of the biological world. Its operation is highly orchestrated, displaying exquisite and intelligent design.
So as we saw in the animation, the topoisomerase enzyme reveals exquisite design. It literally is able to cut one strand of the DNA past the other strand through the gap and then reseal the break. It's an incredibly designed protein, but that's just one of many proteins that are essential for the DNA replication machinery to work. Another enzyme is the DNA helicase, which actually is responsible for breaking the hydrogen bonds along the DNA double helix in order to prepare for copying. It unzips the DNA double helix, if you will. And again, this is an incredibly well-designed and structured protein. Let me show you a short animation of how the helicase enzyme works.
Helicases separate nucleic acid duplexes into their component strands using energy from ATP hydrolysis. The crystal structure of this DNA helicase from bacteriophage T7 reveals a hexagonal arrangement of 6 identical subunits. Surprisingly, the ring is not 6-fold symmetric, but is slightly squished. A model for the mechanism of how the enzyme might work explains this structural asymmetry. Of the 6 potential ATP binding sites, 2 opposing ones bind ATP tightly, 2 are more likely to bind ADP and phosphate, and 2 are empty. These 3 states may interconvert in a coordinate fashion as ATP is hydrolyzed, creating a ripple effect that continuously runs around the ring. Because of these conformational changes, the loops that extend into the center hole of the ring that are proposed to bind DNA oscillate up and down as seen in this cross-section. The oscillating loops might pull a DNA strand through the central hole, thus unwinding the double helix in the process. A frontal view shows the full dynamics of this fascinating protein machine.
Pretty cool, huh? And the helicase spins the DNA literally as fast as a jet engine. It's incredibly, it's an incredible protein. There was also the DNA polymerase that actually performs the copying. I won't show an animation of this, but it's an incredible protein that has its own proofreading function in many organisms. It's an incredibly well-engineered protein. If you don't have the polymerase, then there's no copying that's going to take place. If you don't have the helicase, there's not going to be any unzipping of the DNA for copying. If you don't have the topoisomerase, it's the DNA is going to break during copying. So as you can see where this is going. If you take the single-stranded binding proteins are essential because the single-stranded binding proteins actually bind to the single strands of DNA during copying to prevent them from re-annealing while the copying process is taking place. You have to have the DNA primase because the DNA polymerase isn't able to begin copying without the presence of a free, it's a chemical group called a 3' hydroxyl group, and it's not able to synthesize apart from that. And it needs to be synthesized by a primase, which synthesizes a short RNA primer that the DNA the DNA polymerase can extend from. So if you don't have the primase, then again, no copying can take place. You have to have a DNA ligase, which ligates. As you saw in the first animation, because of the anti-parallel nature of the DNA, where the strands are bi-oriented relative to one another, one of the strands, it's called the lagging strand, actually loops around and has to be copied backwards and discontinuously in order for the strands to be copied in the same direction. And as a result of that, there are on the lagging strand, you have these short RNA fragments or primers that are synthesized. And then from these RNA primers are synthesized short fragments of DNA called Okazaki fragments. And if we didn't have, and the RNA primers have to get removed ultimately and replaced by DNA. And the Okazaki fragments, these fragments of DNA along the lagging strand, have to get stitched together or glued together. And the gluing together is accomplished by this protein called ligase. So it's also essential. So again, if you were missing any of these components, you don't have a system that works half as well as it used to or quarter as well as it used to, but it's completely broken. And so by a trial and error type of process of natural selection acting on random changes, how would you explain a system like this incrementally? It doesn't make any sense. And I would call this an example of irreducible complexity on steroids, because being able to replicate yourself is essential for differential survival. And differential survival, of course, is needed for natural selection to work. And so you can't really invoke natural selection to explain the emergence of a system like this without invoking the existence of the very thing you're trying to explain, namely a self-replicating system. Finally, let's consider the causal circularity problem. So let me remind you of what we discussed earlier, that DNA is transcribed into messenger RNA, which is an intermediary molecule, and then it's taken outside of the nucleus in eukaryotes that have nuclei. So it's taken outside the nucleus, and then it's translated into proteins like the ribosome. Bacterial cells don't have a nucleus. So in the case of bacteria, transcription and translation actually occur at the same time. But it is a technical point that doesn't really make a difference here. So this is known as the central dogma of molecular biology. Now, this presents an enigma, because DNA codes for proteins, but proteins are needed in order to replicate and transcribe the DNA. So which came first, right? The DNA or the proteins, right? The chicken or the egg problem. Another causal circularity problem is the DNA codes for proteins that are made of sequences of amino acids, so as we've discussed. And most amino acids have their own dedicated biosynthetic pathway. In mammals, there's an exception of 9 amino acids, which are known as the essential amino acids, which of course are essential in the diet. But of course, primitive bacteria wouldn't have had that luxury. So most amino acids have their own dedicated biosynthetic pathway, but the enzymes involved in the biosynthetic pathway are themselves comprised of amino acids that are encoded by the DNA. All right, so this is a significant enigma. And I might add that the nucleotides also have biosynthetic pathways that are mediated by enzymes that are comprised of amino acids that are coded by the DNA. So you have all these, causal circularities. And this is a big problem, I think, for an unguided naturalistic account of their origins, but makes much more sense on the hypothesis of design. Now, how have evolutionary biologists sought to address this problem? Well, the most popular contender is what we call the RNA world scenario, which is to say that once upon a time, the chicken actually was the egg, right? In the chicken and egg paradox, where it's been shown that RNA is able to form three-dimensional structures by complementary base pairing with itself, like you see here on the right-hand figure. So this is what we call a ribozyme. And it's able to perform some chemistry. It's able to perform some of the jobs that we might otherwise ascribe to protein-based enzymes. And so this has given rise to the hope that perhaps an RNA-based world, prior to the DNA-protein world, was able to get the job done, and it was able to be a viable alternative to the DNA and protein-based world. So the idea is you start off with organic molecules that leads to the RNA world, and then eventually you move from there to the RNA protein world, and then finally to the DNA protein world that we have today. And all organisms have a DNA protein world today, except for a few viruses, which aren't technically organisms which have RNA coding for proteins, but still an RNA protein world, not an exclusively RNA world. So this is the RNA world scenario. Now, there's a number of problems with the RNA world scenario, which lead me to think that it doesn't actually address the causal circularity problems that we just discussed. So, for example, RNA building blocks are notoriously hard to synthesize and easy to destroy. In fact, RNA is incredibly unstable relative to DNA. It has an extra chemical group called a 2' hydroxyl group that makes it more prone to hydrolysis. than DNA. It's also single-stranded, and so it's unstable from that point of view as well. It's in fact, researchers, when they're using RNA in the lab, they actually reverse transcribe it from RNA into DNA. It's called cDNA, and that's the reason they make cDNA is because RNA is notoriously difficult to work with in the lab because it's very easy to destroy and it degrades very readily. So this is a significant hurdle. And of course, the RNA building blocks are themselves difficult to create or synthesize in the first place. So at the boiling point of water, adenine and guanine possess a half-life of 1 year. The half-life of uracil and cytosine is 12 years and 19 days, respectively. In hydrothermal vents, which is often thought to be the location of the first life, the temperatures would, of course, be much higher than this. Second problem is that ribozymes are poor substitutes for proteins, right? So they're able to perform some of the jobs that proteins would accomplish, but only a limited subset. So, for example, RNA molecules can cleave other RNA molecules. Add the phosphodiester bond through hydrolysis. RNA molecules can ligate separate strands of RNA by catalyzing the formation of phosphodiester bonds. Ribosomal RNA promotes peptide bond formation within the ribosome and can promote peptide bonding outside the ribosome, although only in association with an additional enzyme, which is, of course, protein. So these are significant. These are some of the known functions that ribozymes can perform. But beyond those, RNA can perform only a few minor functional roles, and then usually as a result of scientists intentionally engineering the RNA catalyst in question. So it doesn't, I don't think it really has the potential to substitute for a protein-based world where we have actual protein-based enzymes. A third obstacle is that an RNA-based translation and coding system is implausible. So an RNA-based replication system eventually would have to begin to produce proteins, and not just any proteins, but proteins capable of template-directed protein manufacture. But in order for that to happen, the RNA replicator first would need to produce machinery for building proteins. And so as the first step towards building proteins, the primitive replicator would have to produce RNA molecules that are capable of performing the functions of the modern proteins involved in translation. Also, the RNA world doesn't explain the origin of genetic information. So even if a system of ribozymes for building proteins had arisen from an RNA replicator, that system of molecules would still need information-rich templates for building specific proteins. Ribozymes also haven't been shown to be capable of complete self-replication, right? The ribozyme engineering experiments have only shown them to be capable of partial self-replication. When RNA forms complementary base pairs to fold back on itself and form a three-dimensional structure, part of the molecule no longer presents an exposed strand that can serve as a template for copying. And so thus there's a physical limitation on the capability of RNA to self-replicate. There's also the problem of minimal replication fidelity. So this is from a paper, this is widely discussed in the literature, but this is from a paper by Seigart titled Evidence for Phase Transitions in Replication Fidelity and Survival Probability of the Origin of Life. And he notes in the paper that when the replication fidelity, that is the copying accuracy of genetic material, falls below a certain threshold, quote, modern organisms undergo an error catastrophe from which they cannot recover, as has been shown in the cases of viruses, aging and evolution, and in the macromolecular replication in early life. So, for example, viruses, especially RNA viruses, are close to this critical threshold with approximately 1 mutation per replication. And in fact, increasing the rate of mutations to result in an error catastrophe has been proposed as an antiviral strategy. So you don't have the luxury of trial and error trying to get that minimal replication fidelity in the primitive life. So again, you've got this, you've got this kind of problem of causal circularity where you need to have natural selection doesn't help you to acquire that minimal replication fidelity because you have to presuppose the existence of the very thing you're trying to account for. Now, what's the minimal level of replication fidelity needed to sustain life? Well, this has been estimated to be equal to 1 minus 1 over L, where L is the length of the information molecule polymer. The size of the smallest ribozymes is in the ballpark of around 50 ribonucleotides. For a string of ribonucleotides of this length, Jack Szostak, who's a famous origin of life researcher, concludes that, quote, the error rate during template copying should be less than approximately 2% at each position. But what is the average error rate during RNA copying? Well, according to one study, this was estimated to be around 17%, which is significantly higher than this maximum error rate. So this is a significant conundrum. So in conclusion then, it's first of all, a life-permitting universe at all is unlikely on naturalism, and irreducibly complex systems challenge naturalistic accounts of life's origins, and intelligent design predicts these observations better than does naturalism, and so it fits far better on an intelligent design hypothesis than it does on a naturalistic hypothesis. So I'll end with some scripture. So this is from Romans 1:20. Which says, this is by the Apostle Paul, written in the 1st century. He says, for his invisible attributes, namely his eternal power and divine nature, have been clearly perceived ever since the creation of the world and the things that have been made, so they are without excuse. And of course, Psalm 19:1 says that the heavens declare the glory of God, the sky above proclaims his handiwork. How vindicated the psalmist or the Apostle Paul would feel today in light of our modern knowledge of the incredible design of not just biological realm, but also, of course, physics and the incredible fine-tuning of laws and constants of our universe, and the prior environmental fitness of nature, the fine-tuning of the periodic table to allow for carbon-based life, especially complex life, and so forth. If you want some further reading on what we've been discussing, I recommend these books, Darwin's Black Box by Michael Behe, which is lays out the case for irreducible complexity. It was written in 1996, a little dated, but the essential arguments, I think, still carry significant merit. Foresight by Marcus Eberlin, who's a Brazilian scientist. The Cell's Design by Fazali Rana. And finally, Signature in the Cell, DNA and Evidence for Intelligent Design by Dr. Stephen C. Meyer. And again, you can contact me at, you can find my website, jonathanmcclatchie.com, email me at jmcclatchie@discovery.org, and you can find the Discovery Institute's website, discovery.org/ID. And I would again encourage anyone that's struggling with doubts or questions concerning the veracity of the Christian faith to get in touch with our team at talkaboutdoubts.com, and we'd love to set up a private conversation with you to talk about your questions and doubts in confidence. So thanks for your attention, appreciate your time, and I'm happy to take any questions if there are any.
Okay, well, that was really good. There was a lot there. I'm sure that people will have some questions. We want to be aware of people's time. If anybody needs to go, can just go. And we can just pray to end our official meeting, but we'll stick around to ask some questions and talk about some things if y'all are interested in that. Okay, so let's pray. Lord Jesus, thank you for this day. Thank you for this evidence, and just thank you for the vast amount of evidence that you've given us to show us that you have in fact created all life here, and that you do in fact care about us, and that you do in fact exist. And so we just pray that we can go and share this information with others. And we just pray that everyone has a good week. And we pray this in Jesus' holy name. Amen. So, like I said, if you want to go, you're free to go. But I assume there are probably a number of questions, especially from the biologists and bioengineers in the room, or maybe not. But at any rate, any questions?
Actually, Dr. McLatchie, my bad. Are you familiar with signaling theory?
With signaling theory?
Yes.
I'm familiar with signaling. I mean, what do you mean by signaling theory?
The process at which signals and communication arises through natural selection.
That's a little outside of my expertise, but what about it?
Basically, I think one of the things that you presented in your lecture was something about where does information come from? How does information arise? And I think you may be familiar, but even Richard Dawkins was he actually expertise in signaling theory. He wrote about it, I believe, birds specifically. But signaling theory provides a basis at which information systems and communication between non-minded entities can communicate with one another. For example, flowers send out pollinating bees.
Yeah. So is there a question there?
I'm just kind of wondering how that would play in to perhaps if signaling theory is shown to be a relevant thing within the evolution of creatures or non-minded entities, how does that play into the intelligent design theory?
So I haven't read really any of the literature on signaling theory, so I don't want to talk too much on a subject outside of my expertise. Yeah, so I'll leave it at that until I've reviewed the literature on that topic.
All right.
Well, I have a question. About viruses, has anyone ever tried to come up with like a theory as to how those would emerge from a non-intelligent design perspective?
I mean, there are hypotheses, yeah. So some people, for example, think that they are perhaps they arose as a means of transferring information between cells, for example, or something along those lines. Perhaps they're escaped mobile elements. But yeah, so there are hypotheses out there.
Are any of those viable, or, I mean, I assume they probably aren't, but what would you say in response to people who say that those things, what would you say in response to that theory? That they're just old parts of cells that sort of escaped or something like that?
I don't think it's completely implausible. I don't have, again, I haven't done a deep dive on the literature on that particular subject. But I don't have any particular prima facie reason to see it as being incredibly implausible, no.
I have one more. Dr. McLatchie, you also talked about William Paley's watchmaker
Right.
art. Yes. And then you talked about how was that from The Origin of Species, where Charles Darwin said that if you could show any complex organ, then my theory would completely fall apart?
Exactly.
Okay. I guess the question I have is the analogy that is typically used by apologists whenever it comes to showing intelligent design is using the eyeball, right? And showing how that, explaining the complexity of the eye and asking, well, how did this all come about? But isn't there a theory for how the eye did come about through natural selection of different cells and the process of which those different cells and these different functions came about?
There is. It's not a very convincing theory. I mean, even Charles Darwin put forward the theory in Origin of Species. I can send, if you send me an email, jim@McLatchie@discover.org, I can send you some materials on that. I've even written an article myself on the subject. I mean, you can get all these eyeballs out from various organisms and slap them down on a table and draw arrows between them and say, oh, look, one evolved, one into the other. But I don't find that very convincing unless you can show a viable naturalistic explanation for going from one to the other. And even getting, a light sensitive patch, which is the very simplest form of vision, I think is prohibitively difficult. And I can send you resources that I think show that quite decisively. And going even from a light sensitive patch to a fully developed vertebrate eye seems to me to be implausible. I don't think that you can plausibly go from a light sensitive patch to the complexity that we see in modern eyeballs. It's also striking that the eye seems to have evolved multiple times, several dozen times, which is interesting.
So the eye has, according to the evolutionary theory, the eye would have had to evolve like several, what is it, like 40 different times or something?
Yeah, that's exactly right. Yeah.
Isn't it the case too that some eyes are, how do you say, if a mammal shares the same family with another, their eyes could have evolved from the same base animal, or basically all these little I'm explaining it poorly. That's if a species is within a genus of a different species, right? More than likely they share attributes that that evolutionary path could have come from, correct?
Right.
Yeah. So it doesn't need to be the case that the eye evolves independently, however many times. It just has to be that the eye takes on a very specific, takes on functional characteristics.
Well, it did
unique-
evolve. Well, I mean, assuming common ancestry to be true, it did arise independently multiple times, right? Because you can't trace all of these different eyes to a common ancestor. So that's a significant problem. It's called convergent evolution, which is, I mean, this isn't controversial, what I'm saying. You can find this written about in many mainstream papers, and I'm sure there's even a Wikipedia page about it. I've also written on the subject. So again, if you send me an email, I'll be happy to send you literature relating to that. So I find far more interesting the biochemistry of how vision works. And it seems to me that the biochemistry of vision is way too complex for it to be the product of mindless chance and physical necessity. If you want a good introduction to that subject, Behe has a discussion of it in his Darwin's Black Box book.
Okay.
I get, anyone else have another question?
Go ahead.
Another question I had, and correct me if I'm wrong, but, so with the endosymbiotic theory where originally, you know, a cell basically consumed the bacterial cell to have mitochondria in it. So, if I'm right, doesn't the mitochondrial RNA have a decent amount of similarities to the bacterial DNA and to like bacterial DNA? And if so, I guess just what would be your response to someone who like had that argument as a like refute to what you were?
Yeah, there are striking similarities between mitochondria and bacteria, particularly both have a circular chromosome. Most bacteria have a circular chromosome, just like mitochondria do. And there's other similarities as well. There's also striking differences between bacteria and mitochondria. I also have an essay published at Evolution News on this subject. So again, if you want a link to that, send me an email and I'll send you the URL. Another issue is the fact that most of the mitochondrial proteins are actually encoded by nuclear DNA. Not all of them, but most of them are, which suggests that the DNA, the genes from the mitochondria actually have to have migrated to the nuclear DNA and become integrated into it, and then were nonetheless expressed in the mitochondria. And of course, mitochondria have a slightly variant genetic code. UGA, for example, in mitochondria codes for tryptophan, whereas normally it's a stop codon. And so you would have to recode the mitochondrial genes on their way over to the nuclear DNA. So there are also some significant problems, I think, with the endosymbiotic thesis.
Any other questions, especially about biology and evolutionary biology and those sorts of things? Especially having someone here with a PhD in evolutionary biology, so. While we have the chance. What, if someone, let's say that, well, I had a question and then I forgot it.
Happens.
Man. That bothers me. Anybody else have anything?
I suppose this, if it's the case that intelligent design is the way in which everything on Earth came about, right? Why do you suppose that scientists today, that most biologists and scientists propose mindless and non-intelligent evolution?
Yeah, I think there's a number of factors at play here. I don't like to psychoanalyze people, and I don't think that we can paint everyone with a broad brush. So I'm hesitant to give an answer there because I don't want to make it sound like it's a one size fits all, because obviously there's very diverse reasons why people come to the conclusions that they do, and it's difficult to psychoanalyze them and say that this is the reason why people think this. But I mean, there are some general reasons, such as I mean, there's a naturalistic worldview that's on the line, and a lot of people are attracted to a naturalistic worldview, and Darwinism is essentially the only game in town if you want to subscribe to a naturalistic worldview. So it's not like a worldview neutral hypothesis. I mean, Richard Lewontin has his famous quote, right, where he says that they reject intelligent design or theistic explanations in principle, not because the scientific evidence is stacked against it per se, but because they're committed to methodological naturalism, because they can't allow a divine foot in the door. And I think that that is certainly at play in among many people. I think also the vast majority of scientists don't know a lot about evolution, and I even include biologists within that category. I think most biologists don't, you know, they have maybe a working knowledge of evolution. They know perhaps the bare basics, but they don't really have a deep knowledge of the subject of evolutionary biology because they're not specialists in evolution. And then of the minority of biologists that are specialists in evolution, the vast majority of them don't know about the scientific critiques of evolution or the arguments for design. And of the few specialists who do know about those critiques, the responses to them are just not very good. So the consensus on this matter doesn't really bother me very much at all.
Okay. I remembered what my question was.
Okay.
It had to do with epigenetics. Can you talk a little bit about the importance of epigenetics, like what it is? I think I got a good idea of what it is, but why that is a problem for evolutionary theory?
Do you have an hour? So, yeah, I have a whole presentation on this. So it's quite difficult to go into this in a few minutes, but I mean, epigenetics in a broad sense is simply information beyond DNA that is involved in that plays a role in the development of an organism. And much of the information responsible for the morphogenesis of organismal form actually is beyond the level of the DNA molecule. It controls the organization of tissues into body plans and so forth. And given that much of the information beyond much of the information responsible for the morphogenesis of organismal form exists beyond the purview of the DNA molecule, you can mutate DNA essentially indefinitely with respect to probabilistic resources and number of trials and so forth. You'll never generate the type of form and structure you need to build a fundamentally new type of organism. So there's a response in a nutshell. But if you would like to discuss that more, then happy to speak another time on epigenetics.
So essentially, a lot of the changes that we see that we would attribute to, that would have years ago been attributed to evolutionary changes in DNA, were actually different expressions, epigenetic expressions. Is that fair to say?
Yeah, I mean, the body plan, I think, is not determined exclusively by the DNA molecule. There's higher levels of information beyond the DNA that determine. I mean, DNA can't give you a spatial coordinate system, right? So DNA can determine where a protein is expressed, but it doesn't give you the spatial coordinate system that is above the DNA molecule. And so by mutating DNA alone, you're never going to be able to generate a fundamentally new type of organism.
All right. Well, any other questions? Any questions from the biological engineers? No? All right. Well, then I guess we'll call it a week. I really appreciate you taking the time to talk with us. This is really interesting stuff. And yeah, it was really good. So thanks for joining us.
Thank you. I really appreciate the invitation. Have a great rest of your day.
All right, you too.
Thank you.
Bye.
Have a good one.